Thank you very much. I am Amelia Ellen Radi, an undergraduate biology student at Queen Mary University. And I would like to introduce the next section, which is run by Dr. Rashidat Abdulaziz. She will be hosting the roundtable discussing working with Drosophila in Africa. Dr. Rashidat is the DrosAfrica Outreach Coordinator for Nigeria. And behind me is an image of her outreach activity. And she is a co-founder of Droso for Nigeria and the pioneer laboratory manager of the first Drosophila lab in her university and in northern Nigeria. So I encourage you all to unmute and discuss. And I will pass on to Dr. Rashidat.
Let's see how we unmute everyone. Rashida, are you there? Let me try, maybe we have to unmute her. I'm trying to unmute everyone. I don't see her. Let's see. Rashida, where are you? No, maybe she got disconnected. Rashida, I don't see or hear you.
I will send her an email, Isa, to see what's going on.
Okay, because I saw her before. I don't see her in the list. And I will also try to unmute everyone so you can all participate in the round table. Let's give her another minute or so and see whether it works out. If it doesn't, then I guess it would be okay to move on to the next speaker. Rose, would you be happy to do that if Rashida is not available? Great. Okay, let's give her a minute. See what's happened. So I will wait to unmute everyone once I know that Rashida is connected, because I wouldn't want to have that for Rose talk. Yeah. Okay, she's trying to connect. And I've, in any case, asked Ross to unmute you now. So if you can unmute yourself. Yeah, that's good.
I am unmuted. I haven't spoken in 3 hours. So my voice was a little shocked. So let me know if you want me to pick up the baton.
Yeah. Amos, could you update us with what you're trying to do with Rashida? Shall we wait for her, or shall we move on to Ross and then we can do the round table at the end?
I think we could move to Ross and then
yeah,
do the round table in the end, which is also a good option anyway.
Yeah. Okay.
Amos wrote that he just called.
Okay. So I'll pass you to Amelia then again. Thank you.
All right. So now we will move on to Professor Ross Cagan. And Dr. Cagan leads the Center for Personalized Cancer Therapeutics team. that treats cancer patients through a personalized fly-to-bedside clinical trial. And he is the scientific director of the Wolfson Cancer Research Center, where his laboratory has developed whole animal models for cancer and genetic diseases. So today he will be talking about curing fly cancer. So I'll hand over to you now.
Thank you for the lovely introduction. For those of you who were looking forward to seeing somebody else on this moment in time, you got me. So I'm going to share my screen in the first slide. I'm also still waking up my voice. And the first slide here is a long overdue thank you. Let's see, how do I get this going here? Ah, lovely. Back. So this is a movie from last year because we don't have them for this year yet. Hopefully that will change. I just wanted to thank Isa, Lola, Marta, you can see my screen, everybody from Ghana. You guys have done an absolutely amazing job of putting this workshop together. I have learned a lot and much appreciated for all those efforts. And also, before I forget, I want to thank people in my own laboratory, which I've just managed to get started up, having moved here to Glasgow and then walked right into a pandemic. So Hamid, who spoke beautifully here today, Boji, Shri, Evangelia, Kimi, Tina, I know many of you have been keeping track of this workshop. Thanks everybody for all your efforts, both here in Glasgow and back in New York. Much obliged. So I'm going to need to get on with it. So quick outline, I'm going to talk about two things and try to do one thing that is published, but also some work that's unpublished because I haven't really talked about our work in rasopathies. So I thought this would be a really cool place to really open up and introduce to you all. So I'm going to talk about cancer, what is it, a little bit about fly to bedside, which you've heard about, and our efforts to develop drugs. And I just took out breast cancer because I realized we wouldn't have a chance to talk about that. But we do have a fly model on breast cancer. And instead, I'm going to lean in on rasopathies and then, like all of us, talk a little bit big picture. So I think the first thing we can all agree, cancer is bad. It is in the Western world, it is with heart disease, the major killer of people in the West. I had a lot of data for Africa, which unfortunately, again, I've had to pull just for time reasons, but it's just to put us all on the same page. So Drosophila models of cancer really look a lot like our own. So cancer is characterized by a lot of cell division that creates tumors. These tumors, as we've talked about, can attract blood supply to them. That's a problem. But these initial tumors don't kill for the most part. If I have a tumor in my prostate or my breast, that lump is not going to kill me. It's when cells leave the primary tumor, as you see here, and they pop out into other places, your liver, your brain, your bones, that they really become a problem. So most cancer deaths are due to metastasis. That's when cells leave the mothership and spread elsewhere. And that has been really challenging to treat. So if we take a moment to think about cancer therapies over the years, I have to say I'm about to blow through about 2,000 years of cancer therapy efforts in about 1 minute. But just to get us onto the same page. So some of the earliest descriptions of cancer actually came from Egyptian scrolls known in the West as the Edwin Smith papyrus. These are medical records from over 3,500 years ago. And in those records, it included 8 breast cancer patients and also the treatment of those patients, which was mainly cauterization, literally burning it out, which, considering they didn't have anesthesia, that's no joke. And discussed in those papyrus is how frustrating it was, those treatments. It's interesting, 3,500 years later, it's still a challenge to get these cancers under control. It's really, really amazing how difficult a problem it has been. So there are just a small number of categories of treatments. The most effective by far, even though it's no longer the coolest thing to talk about, is surgery. If you take the tumor out and you get all of it, you're good to go. And most cancers are cured that way. However, the key phrase there, of course, was if you get all of it. And we get back to metastasis. So when cells leave the mothership and start running around the body, this is where we get into trouble. And so this led to, into the 20th century, a variety of treatments, starting with DNA damaging agents, radiation or chemotherapy, okay? These damage the DNA, and then when the cells try to divide, they have a program that checks on the DNA, and if it doesn't make it through that checkpoint, the cell self-kills. So everything that divides your tumor, your skin cells, your hair cells, your gut, and so chemo and radiation have terrible side effects. So more recently, there have been efforts on what we call precision medicine. And ironically, I'm now the Regis Professor of Precision Medicine, and that's ironic because I've been sort of fighting against the idea of simple precision medicine. The way that we think about precision medicine is that you look at a patient's tumor, you sequence it, you say, ooh, you have a mutation in protein X, EGF receptor, KRAS, PIK3CA, et cetera. I will give you a drug that hits that target directly. So how's that precision approach working? Not so great. So today, in 2021, approximately 20% of solid tumors, if you fail surgery and chemo, about 20% of them, we have a treatment that will do anything to the tumor, not cure it, but do anything at all to the tumor. 20%. That includes immunotherapy as well as targeted therapies. So my purpose is not to be disparaging at all. 20% is something, but it still leaves the majority of tumors where once they get past standard therapy, we really don't have much for these patients. So there's a big world out there of therapeutics to be worked on. So that brings us to the fly. As you've heard in the last 3 days, flies, first of all, flies are fantastic. I love flies. If it were appropriate, I would cry right now for flies. So many of the cancer genes were actually discovered in flies, and Eric Wieschaus, who you heard from, discovered many of them, he and Yanni in the screen. Many of the tissues and the genes are conserved between flies and humans. And of course, they have very powerful tools. And I think that Eric said it very well when he noted that one of the things moving forward with flies is they're still amongst the easiest tools to use to get at complicated problems. And I think Michael today has really shown that with the circadian work, really walking through individual neurons. And they allow you to think about what I like to call therapeutic networks. So instead of simplifying things gene by gene by gene, we have powerful fly tools now. You can think about networks. I'm going to get back to this concept. So there are plenty of disadvantages in flies. If I was giving a full-blown talk, I would talk about some of those. But the major disadvantage of flies is that the details can be different. And when you're treating a patient, details matter. So Hamid spoke about the history of flies. So mercifully, I don't have to. I just want to add something to this run. So most of this work until the right side in the red was really focused on developmental and cell biology. And those will continue to be of key importance. But I'd like to add to the points by Michael and by Eric by saying that another world to build on in Drosophila, and a world they're really beginning to embrace, is the world of disease. So one of the major challenges in dealing with difficult diseases, cancer, Alzheimer's, Parkinson's, et cetera, is that these are complicated diseases that affect the entire body and are affected by the entire body. Having a whole animal that you can take these apart, I know many of you are interested in toxicology, same argument. having a whole body tool to take those apart is really invaluable, and that's going to be true for a while. And to push that further, that brings us to areas of therapeutics where you can find therapies that don't just attack the cells that are affected, the tumor, but attack the whole body in a way that can recruit the body to get rid of the tumor. So the bottom line is the 21st century flies have a bright future, and some of that definitely should be in disease and especially embracing therapeutics. So having discussed the complexity of some of these challenges like cancer, let's take a deeper dive into this. So traditionally, thinking about precision medicine is generally thinking about this gene is important or this gene is important. But I think everybody here understands that development, and especially disease, it's a complicated network. So you have the networks of the cancer itself, for example. And so that's represented here in this red labeled network. And precision approach will be identifying one of the key drivers of that disease and hitting it with a drug. And I showed you the data on that. That has worked sometimes, but overall has not been particularly successful. And of course, part of the problem is that when you go after these key drivers, the reason they cause cancer is because they're fundamental to the biology of the cell. And when you drug those out, you're drugging them out in the entire body. That can be a problem. It's what we call toxicity. So cancer drugs, precision drugs, are amongst the most toxic drugs that we have available for people. Okay. But the problem is even more challenging because you don't have to just think about the network within the tumor itself when you're trying to get to it. You have other networks that are being hit by those drugs as well. So you have your kidney networks, which I randomly call in green. You have your liver networks, brain is affected. All of these things interact with each other. And if you were to sort of break the body down into networks, you would realize that getting after these networks is really difficult. And then on top of that difficulty, each person that walks into the clinic is themselves very different from every other patient. So as people in my lab know, we've been modeling specific patients with our flies. Every patient gets a different fly avatar to model its disease. So it's really challenging. And this is where I think simple model systems like DrosAfrica can really shine. So a quick overview of how my lab is trying to get at these. And as I said, I'm going to sort of surprisingly walk away from cancer on the second part of my talk and talk about Rasopathies. When we try to get at these issues of disease, complex models, the record in my lab is 18 different genes altered in a single fly to capture a patient's tumor. We're running a fly-to-bedside clinical trial, as was mentioned. And we also are developing drugs. So we've built complex models for a broad swath of different cancer types: colon, pancreas, breast, thyroid, salivary. I've probably forgotten some. And each of these models is different. So different tissue types matter, but also each patient within each model is quite different. Okay. And here we go. Somehow I'm losing my voice, even though I've hardly used it today. So Hamid mentioned this. I'm just going to show you a little bit of the data. So this is our fly-to-bedside clinical trial. Officially, it's still going, although I have to say COVID has kind of crushed it. So we've published 2 patient reports, and we'll probably have to wrap this up some point soon if we can't treat more patients, which we're working on doing now. But basically, a patient comes into the clinic. We do a full-on genetic analysis. We sequence the tumor, copy number variation. We use antibodies to probe different pathways. And based on that, we build for each patient their own personalized fly avatar. So everybody's fly avatar is different. We then use robotics to do a high throughput drug screen, every available FDA approved drug through multiple rounds. And doing all of that, we create an individualized drug cocktail for each patient. And you can see one example here of a patient that were reported a little while back. This patient was, I believe, 54 years old. He was essentially in hospice. He had very advanced disease. We found a 2-drug cocktail, really unusual one, trametinib plus zoledronic acid. We created a 9-hit avatar for this patient, found that 2-drug cocktail worked for it, treated the patient. Here's the metastasis here, and you can see just a few months later that this and its other metastatic burden shrank. It overall more than halved. This patient lived for a bit under 2 years. These drugs were effective for about a year, but the tumor did eventually come back. So we are having partial success, but we're not curing anybody yet. Also, we put quite a bit of effort into drugs, and actually Hamid is the new drug czar in the room, in the lab. It's by an approach we call chemical evolution that we've developed over the years with our chemistry friends, especially Arvindar and Jin. And essentially what we do is we take an initial hit from a screen, we feed that initial hit to flies, we make 20 analogs of each of those hits, we feed those to flies, we take the best of those, we make 20 analogs of those, we feed those to flies, and slowly, step by step, we chemically improve the drugs. So for example, with this initial hit, 5% of the flies lived in this thyroid cancer model. And by the time we were done evolving the drug, 85% of the flies lived. And this is a compound we're pushing with Arvindar, trying to push into the clinics, actually for colon cancer related compound. So we've actually done this for 6 different chemical spaces. We have lead compounds for colon, thyroid, Rasopathies, I'll get back to, liver cancer, and so on. And as Hamid mentioned, one of the compounds we found early on went into the clinics and was approved for thyroid cancer. So this is a big effort in the laboratory. Okay, so I want to finish, how am I doing? I want to finish my talk by talking about something I really haven't discussed much. We just published our first paper on it, which is our efforts on rare inherited genetic diseases, single hit diseases known as Mendelian diseases, okay? These are typically diseases that sometimes you inherit from your parents, but more often, these are spontaneous mutations that happen in the mother's womb. We have been looking at 2 different Mendelian diseases, tauopathies, which is a type of inherited Alzheimer's, and what I'm going to talk about today, Rasopathies. So Rasopathies are pretty much what the name sounds like. They are due to mutations in the RAS signaling pathway. And these children have a variety of defects, as I'll show. These are some of the people. I want to give special shout out to Kimmy, to Evangelia, and it's been a close collaboration with Bruce Gelb and his group. Bruce is a physician scientist who treats these patients and also works in the laboratory. So Rasopathies, these children have a variety of defects. They have bone problems, so they have short stature. They have some developmental delays. They can have intellectual delays. They have an elevated cancer risk. They have a lot of problems. The problem that I'm most interested in is cardiovascular disease. Their hearts thicken, and that's the major reason that these children die. And the reason that I'm interested in that is not simply because it's the major reason that these kids die, but because it's something we can run a clinical trial on. Each child can become their own control. You measure the heart, you say, look, this heart looks thick, treat them with a drug, come back to the same child 6 weeks later, say, ooh, that heart looks better. So you know that your drug is working. And for these rare genetic diseases, having people be their own control really helps in terms of getting good data. So there are a variety of mutations that can cause Rasopathies throughout the RAS pathway. And we have made now, actually, if you count the controls, 30, but I'll say 24 different humanized fly models. Each fly model contains a different human gene that causes Rasopathies in humans, and they show strikingly similar defects in the flies as well. Okay, So for example, I'll just show you a little bit of data from a single protein, RAF1. There are two different mutations that are associated with rasopathy in these children. So we've modeled both of those in flies. And what we found was really interesting. Even though they're mutations in the same protein, in one case, a mutation here near the C terminus affects the wing of the fly when we express it in the wing, but not the eye or the back, the notum. But a different mutation in the same protein does not affect the wing, but strongly affects the eye and notum. So we see different mutations affect different tissues. And this is exactly what you see in these children. So these different mutations are associated with different problems in these children, and that would all make sense. So we've done a lot of this analysis, and what we find is that although they're all broadly called rasopathy mutations, really each mutation has a distinct flavor, and then we can understand why these children look so different from each other. So what did we do about that? Well, one thing we did was we took 13 rasopathy lines. So this was there to DOS, this incredible amount of work. And he screened all 13 lines with 80 drugs. So 80 times 13. And what did he find? First of all, he found that each line responded to a different set of drugs than the other lines, which is pretty amazing considering they all are supposed to just activate RAS signaling. So every line is really very different. So we wanted to find drugs that would work across many of the lines. We found two. Okay, and the reason we were interested in finding things that worked across many lines is, again, we want to think about clinical trials where we have one drug to treat as many of the rasopathy children as we can. I'll call them R1 and AD80. And I just want to show you that compared to DMSO controls, which are basically 0% survival, you can see that all of these show some survival here in these flies, both AD80 in green and R1 in blue. So this seemed very promising. We looked more closely at the fly hearts. Remember, I care about the heart, or we care about the heart, because this is something you can test for in a clinical trial. So here is work we did with Rolf Bodmer and Karen Okur. And here is an image of a fly heart. Just pay attention to this inner surface here called the myocardium. So kind of the equivalent of our left ventricle. If you express a rasopathy gene, you can see that it thickens just like it does in children. And if we give a drug, for example, R1, we can bring that back to normal. And other aspects of the heart normalize as well, the heartbeat, the rhythm, and so on, really restoring the heart back to normal. We've done similar experiments now in human cells. So for example, in induced pluripotent stem cells, IPS, taken from humans either with normal or rasopathy children. We add growth factors to them. It's the royal we. This was done with Bruce Gell, to create heart cells, cultured heart cells. And compared to these two normal cells, you can see that when you put a rasopathy mutation in, these cells get much thicker than, for example, any of these individual cells. So they're sort of recapitulating the thickening of the heart. And if we add either R1 or AD80, these two drugs we found in flies, compared to this thickened heart here, which is very large, that they drop the size of the heart almost back to normal, and this would be normal here. So this is good. We've done similar work with Maria Contaridis looking in mice. Here's data from within R1. And again, compared to control, the mice show an enlarged heart, just like the children do. And just treating with R1 for a few weeks is sufficient to bring that heart back towards normal. So this is important because this tells us we can reverse the heart problems. So we can treat these children after they have heart issues. And that's good. So we're very excited about this. And we're working with a company now to drive R1 into the clinics. And finally, to finish up, we've done our favorite trick, which is doing drug development through chemical evolution, where we start with an initial hit and then evolve it up. So and now it's Kimmy Stevens, working with Jin, chemist. We screened an initial library of 15,000 random compounds. We got a small number of hits, We took the one that looked most like a drug, which we call M1, and we've now evolved it from here all the way up to here. This is rescue of flies. So now we've got a series of compounds which do a beautiful job of bringing these Rasopathy flies back to normal. And if we go back to those cultured human heart cells, those cardiomyocytes, just as we would hope, compared to control, just DMSO alone, these thickened hearts here, these two drugs that we evolved, both of them do a beautiful job of bringing the heart back to normal. So very, very excited about this. And my last point on this, so do these compounds work in cancer?
Sorry.
Mm-hmm.
Hi, sorry, Professor. Just to let you know that you have a couple of minutes and then we'll just move on to the questions. So.
Okay, cool. I'm just wrapping up.
All right.
So question, do these new compounds work in cancer? And of course, the answer is, oh yeah. So here's that complicated 9-hit model. I showed that we treated that patient with 2-drug cocktail. We could never find a single drug to work in that, but these 2 compounds actually work quite nicely to improve survival of these flies. So just to wrap this up, the next steps is to dig deeper into these Rasopathies, finish the mouse model, and push probably the R1 compound into the clinics. This is already on the books. And to push these M1 compounds forward. We're pretty excited about them because they're a whole new class of kinase inhibitors that we think could be potent for a variety of diseases. And again, pushing those into clinics. So I had a few more slides to talk about some big picture stuff, but I really just want to skip across to here and just say that this DrosAfrica workshop has really been a privilege for me to be part of. I'm really excited to have made friends with many of you across the continent. It's been a real privilege, and I hope to have a chance to watch many of you grow as scientists. We have a lot of difficult problems, and some of us have shown you some of those difficult problems. And we do think that with some smarts and some flies, that these can be really effectively addressed, including across Africa, because honestly, we need help with these difficult problems, and we would very much appreciate that help. So I'm just going to thank both the New York side of the lab and the Glasgow side of the lab. And I'm going to just stop. And if there's some questions, I'd be happy to take them.
Thank you very much. That was a very interesting talk. We have a question first from Siso Malindisa, who says, when creating the fly avatars, do you selectively replicate the genetic profile of the cancer or the whole genome of the patient?
Yeah, no, that's a great question. So the answer is B, the second one. So if the patient has some germline, so things they've inherited that could potentially be impacting the tumor, we do include those. And we find that they really do matter. So you can see why these patients may have developed tumors, because they were born with mutations that are predisposing. But anyway, everything goes in. We don't think deeply about even whether it's possibly driving the tumor or not. If it affects a protein, we put it in. And let me just say, the punchline that we have found is that when you put in just the cancer genes, you get a tumor. But if you put the cancer genes and a bunch of stuff, what we often call passengers in, those generally change drug response. So we think that everything has to go in, and it's actually important. Most people just study the cancer genes alone. We think you have to use flies to capture more than that.
Thank you very much. And another question from Leonard Martin Bermudo. You seem to use the GAL4 system, which most likely leads to high expression of the mutant forms. Are the different mutations also overexpressed in humans?
Yeah, so that's also an excellent question, and I think points to a weakness of the approach. So we are overexpressing things. We generally express them to the point where we get a useful tumor that we can screen. So we're more interested in utility. But there's no doubt that that in some cases we're expressing it at higher levels than you'll see in a patient. In general, when there are activating mutations, they'll also get amplified in the patient. But to your question, we're probably not mimicking protein levels very accurately, I would say, bottom line. We do try to be as close as we can. So for example, if a patient has one of two copies of a gene mutated and it's knocked out, we'll put in a transgene that will knock down the fly version about 50% to try to mimic that. So we try to get close, but I don't think we're perfect by any means.
Thank you. And another question from Pablo. He says, awesome talk. How well does reversal of cell thickness correlate with recovery of heart function in Drosophila?
Yeah, so let me say, first of all, the two correlate closely. So I would say each time that we have made the heart correct in thickness, heart function has also improved and general viability of the fly has improved, okay? So I would say there's a close correlation between the two, and that's not surprising. One of the reasons I think rhythmicity changes is because of the thickening of the wall. So that's not too shocking. And that apparently, that appears to be playing out in mice as well. So we are improving the thickening of the heart, but what I didn't show was data that the heart in general gets better, and nicely enough, skeletal defects start to reverse, and we have some, and I'll leave it at that. So we think in general, the mouse is improved. We're looking at some of the neurological function now, but I don't really talk about it until we have that validated. So in general, everything gets better.
All right, thank you very much. And finally, from Hamid Badmos, he says you have shown several collaborative works. What are your secrets to successful collaborations?
Bringing in brilliant postdocs, because I think that's what Hamid is fishing for. So one of the beauties of working in flies, someone asked, yeah, you, Hamid. Also, you wear that next week, I'll wear a kilt. One of the beauties of flies is that people love working with fly people because we can generate just floods of data. And certainly if you look at my lab, we've collaborated with all sorts of crazy, out of the fly world people. So the papers that we published on the patients had 30 or 29 authors, which included clinicians, physicians, geneticists, and so on. And in general, when people have come to my lab, they generally, with the exception now of the pandemic, which we didn't have a lot of choice, they generally have not had fly experience. They bring other expertise in. I think the real power of, I guess, what we've been doing in the lab is that we've been using the flies and leveraging them with as many other technologies as possible. This, of course, is where 21st century science is going to go. I think the lone lab taking their best technique and beating it to death, those days have probably mostly ended. And I think these large team collaborations are just going to become more and more the norm, which is a good thing. And so, for example, Hamid is going to be working or is working with chemists. He's getting to know them. And I think it's great for both sides. I know I've learned a ton. So it's a little bit of rambling. I would say that one of the key important skills is not skills at the bench, but social skills to be able to reach across and just say hello to a laboratory. I'd like to talk to you. And I probably do that now about once a week. I call up a random lab from around the planet and just say, hey, can we just talk? I just want to understand what you're doing and see if we have common ground. In fact, I just did that 2 days ago with somebody in Australia for organoids. So I think the new world of science, which is really going to be a ton of fun, this is going to be the norm, and certainly adding Zoom to all of this is just going to make it so much easier. So I think y'all are in for a fun run in terms of science.
All right, thank you very much.